wrong title. Okay, to this workshop is primarily we're going to cover a lot of things about works that's going on, part of a larger consortium on data standards, and then data dissemination, how to submit data. Mostly the data will be the key element that is a lot of being talked about. We think this is essential for people and why we cover it. I'm giving sort of an overview of today's session rather than being an individual talk, so it's primarily the work of other people, but this should give an overview of what's been covered. If I start a bit of a history, how the work in standards started, this is primarily the focus of morning session, which is mainly on aspect of standard, and this is got to do with mainly data exchange. But obviously the interest within the metabolomic community standard started far earlier with several different initiatives, several different groups. I just managed to capture some of early starters, but by no means these are the only one. From standardization on metabolomic reporting structure, mainly by NMR, some of them related to toxicology, but not necessarily. The other aspects were carried out. Two more on plant, Rotamsted, RMET, and they were involved many people in each of these consortiums. So if I just mention the name, this is probably they were leading, but that doesn't mean any other groups were not involved. So the list is quite big. Till partly, I guess, the NIH roadmap funding into metabolomics in US made everyone to be more coordinated and involved. The Metabolomics Society was established around the same time, 2007. We had a Metabolomics Journal by Roy Goodacre and others. And eventually, MSI was set up to guide through a couple of standardization work and reporting. And by that, they set up different working groups, which involved long list of people involved in each working group. And on that basis, the suggestion on how to report, how to capture metabolomic experiments. And these are the list of groups into chemical analysis. Sorry, I got this wrong. Biochemical context capturing, to chemical analysis, to data processing, work on controlled vocabulary or ontology, and exchange format. This is where mostly our recent effort will be focused on. I will come back to later, but this is sort of relationship at a time between how they perceived a metabolomics workflow is. So the relationship between how to generate an hypothesis, test it, capture some experiments, reach to the chemical biology and chemical element, how to deal with statistics and data processing, and how they're all interlinked. This led to several different publications, mainly around 2007. So within each same working group that is set up here, from experimental to biology to chemical analysis to data exchange to different publication, and this is summaries in Roy's article in Metabolomics Journal. But by no means these not the only one. There are many other publications, and there are a few applications at the time of this reporting carried out at the same time. Every one of these will end up with criteria and what to report. You can go and report endlessly about how you did your experiment. This is very good, but maybe too much. Or you can give too little. You probably all encountered papers that you did, research articles that you read and tried to reproduce the result, but you were not able because a crucial part was missing or wasn't there. So the key part would be, what is the minimum requirement? How much is good enough to be able to make the data reproducible? Because that's the key challenge. Maybe now we have identification is a challenge, or the immediate challenge, but science is about reproducibility, and this requires reporting and requires access to data. So this is the theme that we're following, making the data available, making enough information available so people will be able to reproduce data. And out of this minimum reporting, they went on how to some core information, or CIMR, core information for metabolic reporting. our publication, that this is sort of has made the basis of some of our configuration and reportings. Obviously, it changed because when it comes to practical or pragmatic, you have to compromise always between how much is detailed enough and how much is feasible enough to be reported. But these are the questions that we really like to open up. I want this to be more, trying to be controversial. The other speakers are there, but please do ask your questions and try to see if how we can together change elements of making data and data exchange be something like the proteomics community is or genomics communities are for many, many years for metabolomics. But it requires everyone's work, not just one group or individual or a service. So primarily, these are the elements that we see why sort of this reporting didn't change except without some exceptions, that there was no way to be able to exchange data. Exchanging data by a mean that is vendor independent, a currency that everyone can use. So you don't have to go into an exchange place and give your yen to pound to euro or this and that. Euro is the best example. Anywhere you go in most of continental Europe, you just use euro. So this is what by a common currency in exchange, we mean something that works. Okay, there will be compromises. You always lose in exchange rate, but it's a convenient way as well. And making places to data be available. I guess one of the first one was metabolite. So it's a bit of a promotion of metabolites here. That was one of the first repositories for metabolomics experiment, not just reference compound or part of a criteria, but for an entire metabolomics experiment where you accept raw data, accept the converted formats, IDs, and anything that you're happy to provide, your QCs, your blanks, anything that makes a data reproducible. And there are examples that hopefully we come up to. You can keep your data private if you're happy, or you're still publishing, you still do some work, or you can share it. There are dedicated links that you can share with your colleague while the study is still private. So we try to look for cases and for demands and try to be as flexible as possible to encourage people make the data available. By no means metabolites is not the only one. NIH, Workbench, and there are others, Metabolomics Chain in Australia. So there are other repositories that you can actually use. We hope this number to grow. The more it is, the better. We should not really, we are participating on our resources to encourage people to submit data, to give their data. And the more resources there are, the better it is. It brings a sort of a competition for us to try to make this better and easier, more flow. And we always get variety of tests. Some people are quite happy to submit data that are easy for them. They have a bioinformatician or someone who is more very computationally good at, because it's still required works reporting. And some don't, some we help a lot. And there are choices for people to do, and they can go depending on how they prefer to different resources. But going now back to standardization, out of these things now required a next step. Now is the currency. Now is in data exchange partly. This is to be able to now work with bioinformatics centers, at least from the funding prospect, how they started, the Cosmos Coordination of Standard, which is a coordination action, a European money, was primarily due to funding reason, partners are based in Europe. But by no means this is a European-based initiative. As I will show later, and as people in the audience know, this includes worldwide collaboration with people in wider metabolomics community, sort of the bigger real picture being that now there are collaborators in Australia, in Brazil, in the US, North and South, in China and Hong Kong and Japan. So there will be wider and wider, and this slide is a couple of years old now, or a year old now. We hope to really increase this, and this is sort of the venue we try to get people come to this place. Different work packages are there. There are a very long list of people. It will take a lot of my time if I want to go to names and people's involved. So I'm just giving an overview of a couple of these work packages, and more details are given by subsequent speakers and how do we go by Collecting data, exchange format, this will be Stefan's talk. He will describe more in details how the work that he's done in his group with other partners on capturing data within other communities, being proteomics community, and how we go by the data depositors, how to try to disseminate information and exchange data between repositories. So this is essentially an overview, which you can see in the next slide as well, but there are elements that will be captured via certain ISATAB format, which covers a lot of it by Felipe's talk. Primarily would be the experimental metadata capturing, and then there will be data formats, the XML type of markup format, most dominant one being MZML. That comes from instruments. All the data that's relevant to your experimental settings or the parameters that you run captures to more biological friendly or biologist friendly data, which is the tab separated. So this is intended for biologists to be able to report how they ID'd, what steps they took, and still be very usable. Doesn't require you can read the format in Microsoft Excel, while this is more useful for databases and resources to capture this information in slightly more complicated format. It's just not for mass spec, also for NMR. There will be similar formats. We try to move from standard format, developed now 15, 20 years ago, to our new version, XML type for NMR. It's capturing how metabolites were identified or quantified, or how did they calculation done, concentration or reported into tab-separated biologist-friendly to same format for biological metadata. Again, these will be, a lot of it will be covered by other speakers, and they have website. Daniel has a poster, 124. So if someone interested, you can find Daniel Schober on slide 124 to get more information about this group of people. And again, there's a long list of people that are involved, mainly in generating this thing. And then we hope this list grows and grows to be more people involved, not just current. That's the ideal that we are after. To overview you ISA from Felipe talk, again, I have one slide, but in detail, give you more description of how metadata is captured from different tools, a variety of application, and different means that ends up with submission to Metabolite, or the format that could be used for some publishers and journals and other data resources. So became a similar common currency, if you like, for capturing data. So once you go through the steps, this could be not only used for metabolomics, but also for other resources if you in an environment that not just do metabolomics, but other omics as well. To link to some data analysis packages. So RISA links it to R, for example, or Galaxy. Again, his talk, and if you have more questions, would be Felipe's. We try to be more even inclusive, so there are not just that. There are other tools developed by GOM with partnership with Bordeaux University, EXML. So to capture in a storyboard kind of theme. Unfortunately, Kenny couldn't make it. He had a difficulty coming to Japan, so he's not be able to give his talk. But in a way, it's another way to capture, follows the storyline. So people are familiar how they did the experiments when they started. You have your own lab books, a way to capture. So this follows the different ways of capturing information. Eventually, the job of converting would be to us to make it exchangeable, available, as long as people are happy to capture the metadata. This goes further to via work packages. These are, again, work package leaders I mentioned, but obviously there's a long list of people involved, and this is external. This is a complicated slide. I don't want you to really go into detail, but this is mainly the message from this slide is via different resources and repositories, one being Metabolomics Workbench, find a common way that we can exchange data. So we make sure the data went through and processed and captured could be used and changed if some service goes down. There's a lot of history in proteomics community and others that there were web services or metabolites or repository or database that is no longer anymore. If we would have made it more that we were compatible with one another, then transferring data would have been easy. So this is in one of the initiatives, mostly now being driven by Work Package 5 in Metabolome Exchange. I'll show some slides for that, as well as other means to capture data and being able to use that. It's involved publisher and publications. There are already a number of publications and publishers involved in making this data exchangeable. So there was a meeting recently in EBI, and most of the database and resources, whether it's a reference compound, that is the one that houses a particular spectrum or mass spec from a compound, to repositories, which includes all metabolomics, try to work out how we can exchange data and how we make this data available. We try to make it easier for people to access data. It's kind of like a roaming. You roam, you use your phone around the world, you don't know how it works, and there are people around who actually make it possible. So this setup was Metabolome Exchange. You can find it at this .org address. At the current state, there are 3 repositories which they made the data available via feed to Metabolome Exchange. Is Metabolites, Metabolomics Workbench, and GOM. So these are the public data that are public, not private data. And so ideally, we hope that this will be a place for people to search for multiple experiments, or sort of programmatic access for people to be able to make more, look for larger, different type of experiments, and where different data sets are if they're looking for it. So it will be like a Google of current state. So you use Google to find. And I'm meeting tomorrow in time. For example, if you type Arabidopsis, now you can get about 11 data sets from Metabolite, from Metabolomics Workbench, and GOM. This could be also in metabolite level. So you can look for a metabolite and far away this alanine, which people reported on what that, which we discussed this and we work. This is my final slide. So it's not just infrastructures, but we try to link it to, I keep pressing the wrong button, but not just metabolite infrastructure, but to other infrastructures, that are involved in capturing biobanks, for example. This will be the overview of Cosmos. So at the level will be the exchange format, whether it comes from proteomics or mass spec or metabolomics, make it convertible, make the linked data sets available, data repositories available via this common flag. And this is sort of my acknowledgement. I probably want to move quickly to Stefan, and if you have questions, this will be repeated, so we can have the question via this talk. So Stefan, if you want to come up and... If there is immediate question while we're setting up for Stefan's talk, if there's any quick question that I can answer, please. Yes, Rima.
For these wonderful initiatives and the dream trials that was mentioned by Dr. Oliver Fien, just as an advocate of the metabolomics community, how do we get the voice of the community to participate, to send feedback and comments to the people who are building these initiatives? Because at the end, we really want to have, whether it's databases or standards that are accepted by the metabolomics community at large. So beyond having a meeting once a year, how can we get a dialogue going and more effective interactions?
This is a very good question. This is why we are here, primarily to advertise for this. Each one of these initiatives has a working group email. So there are one for exchange format working group email. There are one specifically for NMR working group email. There are ones that are for ISA, and there are ones for data exchange and metabolomic exchange. So these Google groups we set up, so this is an advertise. We probably will, if you want to come and find any of us, or me, or I try to set up a list that you can subscribe to this Google mailing list, and mailing list is one aspect. So, but any other means that people are happy to participate, we are happy to set up. Jules?
Sure. Thank you. Just to answer Rima's question, as part of Cosmos and also the Metabolites initiative, there are 2 curators there. And I know that Razor fills in as a curator from time to time. So my message would actually just be, if you wanted data standards changed in terms of how they're reported, just in a friendly way, make yourself a bit of a pain in the backside, if you like, and go back and say, no, this isn't right, and this isn't right, or actually this doesn't quite capture the experiment that I've done, and I'd like to have this information captured. So while we've got curators that will go in there and interact with people that report the data, now's the ideal time to have those extra fields sort of put in, or feed back to us and say, this bit of information doesn't make any sense at this stage. It makes a lot more sense at other stages. We are trying to define also sort of standard sort of captured formats for common setups, like, for example, the Biocrates kit and things like that at the moment.
There's a lot of work, but I guess we can put that into more discussion afterwards. If you have any more questions, it would be nice to have it in discussion. But for the time, for now, I want to hand in to Stefan to present it. Because I have to keep on the. I think it's mine.
Okay, so welcome to the data standards workshop here. I'm going to talk mainly about mass spec raw data standards in metabolomics and give a bit more of history. Those slides are also available on SlideShare, but I also learned they will be available from the Metabolomics Society website. So we've seen the workflow in metabolomics a few times, and if you go along from sample processing to the instrument, data acquisition, and then some strange data processing up to the interesting bioinformatics, then this is accompanied by the data capturing. And here, okay, good. So these were the standards that Reza has mentioned, and this is the part about machine data, instrument data, that I will give a brief introduction today. And then also the data processing should be accompanied by capturing what you actually did in there. So this slide is not only about the IPB machine park, and we have in the meantime thrown out some of these instruments by now. But it's mainly because when I started at the Institute, there was different instruments out there, and we were developing software for mass spec processing. And I told everybody I'm not going to touch vendor formats because they are a moving target, difficult to process, gets you difficulties on Linux and so on. So I said I want to have the data in an open format so that I can read it with whatever programming language is out there. So the grandfather of the open data formats is actually NetCDF. And what people hardly know is that NetCDF is not really targeted at mass spectrometry. It's basically a container for large amounts of binary data. And then there came this ANDI-MS specification how to encode mass spec data into the CDF container format. Works fine for GCMS, simple LCMS. It's widely supported in software and programming languages. Problem here is it does not really allow you a mixture of MS and MS spectra. And the more complex and the more interesting your mass spec data gets nowadays, the more problems you will have with NetCDF. Another problem is that it has very poor metadata, and I will show an example there. And finally, if you want to read more about that, it's defined in the standard 19 whatever here, which you can buy for $42 at the ASTM. So why you have an open standard that has expansive specification is beyond me, but sorry. So if you look into a NetCDF file, and probably few of you have done that. Actually, let me come back. Who of you has data in mass spectrometers in NetCDF format? Hands up. That is a dozen, 2 dozens, probably 2 dozens. Okay, just wanted to know. Okay, so when you were to look into that, this is the metadata that is in there, and really there is very little specified. You only know this is from a SciEx. You don't know that it's from a Q-star. This is supposed to be a normal phase liquid chromatography, which is wrong because it was in a reverse phase, but the mass spec doesn't know about that. There is some positive polarity. Those here that start with a test are actually not really specified, so it's kind of strange here. And if you go further down, then you will see your actual data is in the NetCDF. So the next thing was the MZ data, which was done by the Proteome Standards Initiative. So here we have some cross-pollination by the proteomics people. And it includes the raw and the measured data, what equipment was used, software, and so on. There was a fairly good vendor support from Bruker Applied Biosystems, and I probably missed more than I actually covered here. And at the same time, there was the competing format MZXML, which was done by the Institute for Systems Biology, if I remember correctly. So this is what such an XML format looks at if you look at the specification. So this here is an open specification. And then people discovered that if you have competing MZXML and MZ data, that this doesn't really work pretty well. So in 2006, the two came together and they worked for quite a few years to actually come up with the successor, the MZML standard, which is today the recommended standard from the proteomics people, and would also suggest that this should be the recommended format by the metabolomics community. So it's more stable in the definition than MZXML. It's better defined than MZ data had. There is reference implementations, I'll show a few, and we also had in the specification process very early vendor involvement. So there is also pretty good vendor involvement. So the high level architecture of such a ZML file is that you have a header that has some metadata and information, and then down here in the run, there you will find the actual spectra. And what is new, we also cover chromatography or chromatogram list, which is important for MRM and targeted metabolomics, for example. So I'm not going to talk you through that, but this is the header that you see here. There's controlled vocabularies, so you disallow people using POS, +1, or positive, but they are all referring to the same thing, meaning positive polarity in your ionization. It has some more, where the file comes from, what type of file it was you converted from. And if you go down further, you see that there is a software mentioned and a specific version, and then what has been done, software, the data was exported, it was recalibrated, and then, it was peak-picked, recalibrated, and then converted to MZML. And this is all captured within the actual software. And when you then go down, you will find your spectrum. You see that it's positive, an MS2, centroided. The selected ion had a certain M over Z. You see that it was 15 electron volt collision energy. And then down here, and I truncated that, down here you see the actual data. This is not human readable anymore, because human readability would have quite some problems in here. So this was how an MZML file looks like in a nutshell. So one of the early implementations for that was OpenMS, originally for proteomics. It reads data in many formats. There we see another problem of NetCDF. It didn't back then work on 64-bit machines, which are fairly commonplace now. And there's a lot of tools that can process the MZML data. A different one is ProteoWizard. Also, again, from the proteomics world, it is open source, just like OpenMS, also supporting many open formats. And the additional thing is that if you run that on Windows, then if you have the vendor libraries installed, you can directly read the vendor formats. And this is the kind of moving target that I myself was trying to avoid. With the help of the ProteoWizard and this MS-Convert tool, this is actually, if you want to submit to metabolites, and if you're asked to do that in MZML, then you would probably use MS-Convert to go from the vendor format to MZML. The people at the Scripps who created the XCMS Online, they have created a very nice help file, which was actually meant to inform you what raw data formats XCMS Online accepts. But what I find most interesting is that they also have a few notes on how you actually convert your data. And for example, if you have a Bruker instrument, then if you want to have the calibrated data, there is a pitfall, and I would like to mention that, it requires a certain registry key on your Windows computer to be set to actually use the recalibrated spectra. This should be usually set upon installation, but it could happen that it's not. So we could have two different computers giving you two different MZML files, which is kind of a problem. For waters, there is no support for the calibration in the waters DLL used at the moment by MS Convert, so that is a problem. You don't get right away calibrated data. The water software data bridge writes NetCDF data in calibrated version. And there is some kludges how with some converter here that requires the instrument software to be installed, which I don't have. Then you will be able to use the calibrated data. But we found that the logMAR scans are intermingled in there, which is not really coming from your samples. So that could also be quite a problem here. So yeah, there is, if you're in the computational side, and that was also mentioned during the workshop on the software development, there is a lot of libraries out there that can take your responsibility of reading and writing MZML off you. So I only have a small selection here. There's stuff for Python, there's stuff for Java, OpenMS and ProteaWizard were mentioned before. If you're in the bioconductor world, then there's the MZR package, which is the underlying data import-export for XCMS, for example. And there's probably many more which I missed. Any developer of a library I missed in here? So if so, please approach me and I put you for the next version of these slides on this list here. So I mentioned Bioconductor because that's a very nice pool of software and packages for Metabolomics. Out of the 500 that were current last year, there is quite a few that work on mass spectrometry data, some of them for proteomics, some of them for Metabolomics. And we have been involved in the development of XML, which for quite a while now also will read the MZML files. Let me ask another question. So of the people who have seen NetCDF data, how many of those have also exported already and used MZML? Reza, do you have eagle eyes? There's one. I see one. We want to see more of you in the future because it really can capture the more important, interesting data. This is one example where there was a very nice bioconductor package done by other people, and we integrated that into XCMS so that you're also able now to use this particular package on all the data formats that XCMS can also read in. And that, I think, was quite a boost of adoption of the mass spec wavelet package here. So the MZR I mentioned is the underlying data import-export for Bioconductor, and really it reads all the formats that we have here. And I will talk about MZ5 in a moment. And we currently have a Google Summer of Code project, which was very interesting because there was somebody who said, I would like to contribute something, what can I do? And we then had a few suggestions what work he could do in MZR. And that is organized mostly by Laurent Gatteau, who is in Cambridge and one of the co-authors of the MZR. So another thing I would like to point out is what is so cool about MZML, and that is that it was from the outset designed for mass spec data, and it had LCMS in mind. But then a number of people came up and said, well, we have imaging data. Imaging mass spectrometry is very cool. So what data format should we use for that? And really, they used the MZML, they called it imaging MZML because it's the same old MZML, but because they had huge data files, they just extracted some of the data that I showed before into a second data file to come over the limitation that sometimes the XML is known to be rather slow to parse, and if you want to go at that particular spectrum, then it's not really blazing fast. But the main part is that they really keep the same idea that we had in MZML. They keep all the ontologies. And in fact, they basically only put an X and Y position onto the spectrum in the XML to show where a particular spectrum actually comes from. And with those open formats, we can, this is not the OpenMS software, but in general, we can do open mass spectrometry imaging software. So if you're in, how many are in imaging mass spectrometry here? 1, 2, great. So if we meet over coffee or green tea, which is actually very nice here, then we can talk about that, unless you're already using it. Are you already using that? Okay, we'll talk about that later. And I also already mentioned the MZ5. And again, this is trying to overcome some of the smaller limitations of XML being slow if you really want to zoom forward and backward between the spectra. And what they did is kind of NetCDF meets MZML. So they put all the advantages of the two formats together. HDF5 is the big cousin of NetCDF. It doesn't care what data you store. And if you have the proper specification, and that's the MZML specification, how to annotate stuff, then you can store mass spec data in that. And you get size reduction, speed increases. It's in the Protea Wizard, MS Convert, fully integrated. You can go forth and back between the two formats. And also from R, from any of the languages, you will have access and libraries to this format. Problem is the files are not human readable anymore, as I showed on the slides before. And while I can have a look in WordPad at the MZML files, you can't do that anymore with HDF5. So there is the size improvement, speed improvement. So they've published that from the Steen lab, and that's also something very cool. And if you're doing mass spec software, you may want to have a look at that. Changing gears, you've seen the slide before, and I very briefly only would like to mention that we are borrowing the concepts that worked in mass spectrometry and would like to forward them to NMR. So basically, we're creating the NMR ML format. So this is 10,000 feet view of an NMR ML file. It has the schema. The schema in XML world is the definition of what you need to describe. There is a controlled vocabulary because you can't define every single pulse sequence here in such a schema. You basically say, we describe the pulse sequence, but again, just as in the positive example, positive pos plus one example for the polarity, you take the actual value from a controlled vocabulary. So that's much more dynamic in the end. We do have converters to go from the NMR vendor formats. We have a validation suite, which is more important for people writing the software to tell you if the outcome is a valid NMR-ML format, if all the required information is in there, and if it's in the right place. There is a number of the Cosmos partners that Reza mentioned involved in here. So Rima, if you want to get in touch, there should be enough people out there who know somebody who is in Cosmos, and if you want to complain right away. We take it as constructive contribution. So infrastructure, you've seen that we are developing the whole thing in the open. It's developed within a version control system. You can see what we had in there last week or 1 month ago. There is a tracker tracking the discussions, what we need to change and look at. The free for open source projects only means that the platform that we used here, the GitHub, is free for open source. Of course, NMR-ML is free for whoever, including non-open source stuff. So you see what contributions have been done. Here are the issues we are currently discussing. So that was a very good choice of the platform, how to host the development of NMR-ML. There's an ontology very similar to the mass spec that I mentioned. It's also based on earlier work, so we didn't invent everything from scratch, but we took what works out there. People from Birmingham were involved in that. There were people from Canada involved in that and still are, and so on. This use of the ontologies that we have mentioned here, which is like controlled vocabulary, categorized. That can later also be, and probably Felipe will be talking about that, that can also be used in the ISATAP format. That will come up then. And also if you think about semantic web technologies, then the ontologies that we create here within the NMRML world will help you to describe experiments in the semantic web. The poster was mentioned before, that's number 124. I didn't manage to put the number up here in time. So please go and see Daniel's poster and he will be able to talk you through that. And I promised in the title a plea. Originally in the program it says a plea to the vendors, but it's actually not only to the vendors. To the vendors we say, please start, or if you already do so, please continue to support open data formats, because in the end it will help you sell more instruments if there is more people out there who have more diverse softwares to actually work on the data you have in your instruments. So it's for a mutual benefit. To the people doing computational mass spectrometry, please use the joint I/O libraries that I mentioned for whatever platform. Don't roll your own, just improve the existing one so we have a central point of good support for the open formats. And if you're biologists, if you're the users of the open format, please start or continue to request open format support when, for example, you buy a new instrument. If you say we need MZML support, then maybe some of the vendors will think about that. And especially in the MS world, we are right away at the point that you should really request that because it's working. Okay, and with that, I think I'm done and looking for your questions.
Any question? Yeah, Lloyd.
Should I wait on the mic? So many of us do use CDFs, and you were very critical of the metadata that's in the front of those, but I believe that's editable, that you can text edit that. Do you see that there would be a value in trying to go back and add metadata to these CDF files for more generic distribution, or you think it's just waste of time and should move on to MZML?
I would be asking the question, would you really do that? If you submit a study of 200 samples, would you go back and edit the metadata that is wrong because the instrument didn't know what kind of chromatography you were using? Would you go back and add that it's coming from a Q-star instrument? Maybe other converters put in more metadata, but I looked at a few, especially GCMS data files, and there was very little. I had on the slide non-existent metadata until I encountered this Q-star example, which had at least some.
So we're required to submit metadata for the most part in some form or another. And so we have an Excel sheet, and you can just copy paste possibly into the CDF file, or does that type of thing not work? And again, is there no value in that, or is it?
What you're describing probably fits very well into the ISATAP, which Felipe will be talking about. So the kind of distinction is that these are the raw data formats, and it really shouldn't be touched by a human. It should be exported properly by the instrument software, and then you can process it with whatever else. So it's mainly here for the software developers to improve the data that is put into the raw files. And then later on, if you describe your experimental metadata in the ISATAP, then that will be cut and pasted from Excel or your LIMS system or whatever. Anybody here, so let me ask a question again. Anybody here who tried to convert their vendor data into something open and failed because it was too complex, because the software was uninstallable, because something else? So for some instruments, you have the vendor exporters. So I know Applied Biosystems has one, Bruker has, I think Thermo exports directly to that. And then you have the open source ProteoWizard alternative thing. And I think the most fascinating thing, they really managed to get the license to redistribute the vendor DLLs as part of the package to make the installation easier. And I think this is the best achievement, not going to put down the other ones, but this is incredible to get that done for redistribution. Okay, if there's nothing else.
Just one thing I want to ask or re-emphasize that I am happy with the softwares that I have, and why should I convert it into, maybe I want to re-emphasize this, why should we reconvert into exchange format? What's there in it for me?
Actually, given that you're standing over there, possibly that metabolites could be asking for open formats. So I've seen a few studies in there that do have raw files, vendor raw files. So you could have in your submission handbook that you only support NetCDF, MZ, something, I don't care what, open data formats. So ball is on your side.
It's like a tennis game now. Okay, I guess. Once more, thank you, Stefan, for his talk. And I guess while Felipe is getting ready, so these are the elements that, the common currency, the exchange format, and then linking to databases and resources that this information could be easily harvested and used. But now, Felipe would talk about more how to capture the experimental side of metadata. So, Felipe.
Thank you, Reza. Good afternoon, everyone. And thank you for the organizer for having us today. So yes, I will take a step back compared to what Stefan has mentioned, which was more closer to the raw data. I will be telling you about how to capture metadata and tell us what the experiment you have carried out is about. And I think I can do one thing. I can follow up the trend that Stefan has set up. I will ask a question myself. So how many of you have published this year or the past 2 years in metabolomic data? Okay, so that's a good number. How many of you have submitted to a repository of metabolomic data? Okay, that's about 20% of the previous number. So I think, and I'm only for this room, I guess that it would be interesting to have the survey on the other side of the system. So in a way, we'll try to expose you to help you as much as we can by explaining you how to do this in the session. Of course, in the 20 minutes that are allocated to me, I cannot go over all these elements. So it will give you a bird's eye view of the project, really. So, but everything that is about the talk today is about the notion of reproducibility, and this is the tenets of science, really. We need to get the data. Publishing a manuscript is not enough nowadays. It was maybe okay 15 years ago, but now we have the means to store the data and to make it available to people for re-evaluation and for the benefit of the community, really. So this is an essential point. And for us, for the community, it is to build a GEO or an REXpress for metabolomic data. And just to toy with the idea of playing with 25,000 study of metabolomic studies in mass spec or NMR acquisition, that would be really interesting, and it's down our responsibility to push the data publicly. So really the key point is to move away from the scribbles that we put on our web lab book than to the nirvana, I guess, of semantic web as it is sold right now. But in between, there is this kind of, mid-layer that we have to live with, and this is the world of some kind of spreadsheet where we hold the metadata. R calls it phenodata. We call it isotab somehow, because we provide a number of tags that allow to keep the data, the metadata about your experiment in a specific order. So this is the environment. We are talking about how do I exchange the study plan, what did I do to my samples or that I've collected for my clinical study or otherwise. And this would be covered by the isotab format. Stefan had described the MZ and MR/ML formats that are being developed and pushed forward. And of course, when it comes to the identification and the assignments, there are MZ tab and NMR tab format. So really, if we look at the metadata, that is the description of your actual data, you could go the very lazy way, which is everything is in the file name, dude. So basically, if you can tell me, if you're good enough to tell me what this experiment is about. And this is usually what people do. They've got nice naming conventions, but just wait a couple of years, and basically you are in trouble. So basically, we need to tag and to categorize the metadata in better ways and to keep a label on things, essentially. So this is what we have been pragmatic with ISATAB. We developed, okay, how do we move from the graph of the experimental design and help people to manage this information, something they're not too keen to do because it's a bit boring, but in tools that are not always there. So we thought, okay, maybe using a spreadsheet-like table would be the tool, would be a way to start structuring information. And this leverages existing format, the MageTab format that has been used by RegExpress at EBI, and also we provide mapping to Pride XML and SRA XML for sequencing application, which means that you can use the same format to track a number of information in the metabolomic world for your experiment, but also it doesn't preclude you from using it to track data using other techniques. So basically, I will tell you now quickly, basically essential. There are 3 files in the ISATAB in the ISA archive. One is the investigation file that gives you the kind of summary, executive overview, where you will tell us why you did the study, how you acquired the data, which are the protocol, the methods, and the type, the purpose, the objective of this protocol. And then a very important thing that we insist on that people should report, especially if they are in the context of an intervention study, they would have to tell us exactly what are the predictor variables or the independent variables that they are manipulating and challenging the system with. And then, of course, information about who did what and who funded the study. The other file, the study file, which is one type of file in the ISA archive, allows you to collect all the information about the samples you have accumulated in your study. So tell us whether you work on yeast, whether you work on human, on dogs, or on plants, and tell us the detail, the genotype, and things like that. Finally, we have several tables that you can define to tell us what kind of data acquisition you've performed. And here we point you to very simple structure. We tell us the input to the assay, and you tell us what kind of data file has been produced, the raw data file. And if you process it, you'd point to a derived data file, which is basically the result of this data processing or data analysis. Again, it could be pointing to a data set that has been filtered, cleaned up, normalized, and then you could also point to a file corresponding to identification. And then finally, if you do differential analysis, for instance, or enrichment analysis, you could provide us with another table. So how do things look? It's very, as I said, it's a table, and for which we have different tags. So this is an example of, quickly, a study sample file where I'm just declaring I'm collecting this sample. It's of type, the organism is Rachis norvegicus, and I'm providing information on the strain. So this tag characteristics allows you to qualify material nodes in the format. Moving on, now I'm applying a protocol to that animal to produce a new sample. So I use the protocol ref tag, which I can qualify with a parameter value tag to report, for instance, the housing condition or the kind of feed I gave my rats. So all these kind of information are especially important when you do metabolomic studies. If you fail to provide this, of course you could see things, but maybe these are confounding factors that could explain some of the variability that you observe, and it's important to capture that information. And again, I mentioned how important we found that the declaration of the independent variable were. So basically, ISATAB format has a specific tag to declare those variables and provide a vector detailing the treatment conditions. And again, this is a very important aspect, bearing in mind how important the experimental design is when reporting your studies. So quickly, I think maybe these slides are not easy, but just finding the last type of data of tables that we are dealing with. Here, I'm showing patterns for reporting LCMS elements. We split when we are looking at different ionization mode or polarity mode during acquisition, we split two assays where we will report information about the performer and the date, again trying to track the information that could be used for identifying confining factors, also information about the chromatographic column or element. In the case of GCMS, we would like to provide information about derivatization, so either these are sample preparation steps that could be reported as protocol steps, and so on. Here, on these columns here, you identify the assay names, and you will provide the raw spectral data file, either in MZML file or netcdf file. I guess that Metabolite team will be able to deal with that. And this is, again, a similar pattern that you can observe for NMR reporting data, where you tell us the kind of acquisition you performed using controlled terminology that could be reported in the format, and provided the file, the pointer to the data files. So, of course, a format is one thing, but it can only be alive if there is an entire infrastructure to support it. And this is basically a quick overview of the different tools that we have built around the format. And all these tools are available, open source, free of charge, completely. You download, and you've got everything available to you. So really, we are, in a way, enforcing or following the recommendation that were edicted this morning, which really try to make everything open so people can join and engage with the community. The more, the merrier. And this is, I think, a very sound framework to start. Instead of reinventing from scratch, you can contribute to this base code and focus on the science. This is what you are most interested in. So, in a way, we have to deal with a number of very diverse experiments, and we do that by creating configurations for the ISA tools, and we have been implementing ISA configurations based on the CIMR recommendation for a number of elements. So we have configurations now for in vitro study, where you see we reuse the basic tags of the ISA format characteristics, but then we have this kind of soft configuration where we add tags, elements to capture key information about the organism, the cell, the kind of life stage we are in, the genotype, and all information in this element here related to the cell cultures. So again, the value of this is when you accumulate, you don't have to be lazy, you can be very specific, and that should be the best practice when reporting data sets. And of course, we've got specific configurations for plant studies or clinical studies. Everything is available on GitHub again. Just check out this place. And if you have comments, please do tell us when we are wrong. Do tell us and interact with us. Tell us where things are not good enough so we can write specification and improve the software for the community. Of course, we are listening. I mean, we listened to the first configurations. People said, well, I can't find my terms. I would like to talk about targeted profiling. I don't have that. So we are refining those configuration and creating new tables so people can be confronted with things they are really comfortable with, and they recognize the different system. And we are also making efforts to move towards areas which have not been tackled so far. So we've been working with Professor Marta Cascante, and maybe she's in the room. Oh, yes, she is. Hello, Marta. So yes, to do more on the metabolic flux analysis. And I think there are here more efforts to be done to support the technique and to support this kind of important experiments. And maybe in the coming year, we will be able to deliver something more stable on that. So quickly on the tools, this is, again, everything is on GitHub. This is the editor for the format, a standalone Java tool with a QR code generator, a spreadsheet-like interface with ontology access. So basically, we access control vocabulary, repository portal. We have visualization component and PubMed search, and also experimental design-based wizard, so which allows you to create templates based on the information about the experimental design. This is basically the look and feel of the interface. As I said, it's very much like Excel, so people are comfortable. However, we have the ability to link up and access vocabulary either by pick and choose or by tagging. The function is provided by the NCBI bioportal. And of course, we need rewards. So we are able to generate automatically from the information that has been supplied to us a number of images, almost publication-ready images of the experimental design and the overall graph. These are dynamically generated. These are glyphs which are populated based on the information that you supply. So really, and tells a story. The graph also allows you to detect possibly irregularities in your overall representation, so maybe indicating mistakes, so you can review the entire process. Finally, we have APIs, so you can interact with the tool. Again, you have more information here if you want to learn more, or grab me after the session. This morning, Dr. Atsushi Fukushima presented an interesting metric R package, and I think it was very, very important for us because it would be nice for us to recommend the metrics package from our ISA package, which allows you to perform I/O operations on the format directly. And basically, you load the ISA file through the R package, creating a Fino data object, perform your processing, but the package also has function to write back to the file. So you can perform all your analysis and processing in R, and then persist that back to the file, making it a kind of smooth tracking of the metadata as you go along. Everything, again, is open source. You can access that on the bioconductor and contribute. And what we have as well is that we have the ability to recommend a number of packages. We have done that with the XCMS. When we load a data file, ISA recognize, OK, this is a LCMS, and there is some NetCDF file. We can say, maybe you should be using this package. And we would like to expand that. There are, of course, a number of packages in R now to support metabolomic data sets. And Batman R, for instance, is another one for NMR data sets. So among the tools that we have also reported to support the format, this is a new component, which is the ISATA viewer. This is for the web, in a way. What you have to do is just, if you want to support a format or build a repository or serve the ISATA files, you just simply reuse this component, and it will be displayed, rendered as such in the ISA file, allowing to view the content of the metadata with no extra effort. So just plug and play now, very easy. One thing that we, over this last 2 years working with metabolites and starting refining, listening to the input, the feedback from users, was the fact that there are issues and we need to document more patterns on how to code information in the format. And one thing that we came out as well is that we have to pay attention now to the semantics of the annotation that we provide. Of course, we could be dirty and provide, in this example, OK, I've got 2 sources and 2 QC runs on this spreadsheet. The first samples are annotated, one with Homo sapiens and liver as a sample, and the other one is musculus and spleen. And I was lazy, and I was, OK, QC, I run a QC just to check if my machine is behaving properly, and I run vanillic acid and water as reference compounds. But then there is a problem here, because obviously, if I look at this coordinate here, vanillic acid is definitely not an organism, and reference compound is definitely not an organism part. So basically, we need to do better. We need to expand the characteristics here and move this information to the right tag in this matrix. Of course, it comes at the cost of a sparser annotation, but it's accurate. And this is the most important thing. And why does it matter? I will show you the next element. You remember one of the first slides, I said we were moving from the wet lab books to this kind of perfect world of linked data representation. And this is key, basically. What we have been doing is working on the conversion from the ISA tab format to ISA RDF and OWL using this component that we are, again, releasing open source on GitHub. And basically, we can now query a Sparkle using the Sparkle endpoint, all the data set that we have downloaded from Metabolite and converts it into a RDF graph. At the moment, we have performed about 80% of the conversion of these data sets. We can really start doing interesting things, such as creating cohorts. We can cut and slice across different studies and retrieve all the liver samples across the elements, for instance, or all the plasma samples. We are working at the moment also on converting the identification elements, so the MAF file to RDF. So this is the metabolite assignment file to RDF, which enables query from experimental data to chemical entities and vice versa. But this is where also the curation rules will have to be tightened. And this is an important point for everyone if we really want to allow the flow of information to go across the different systems. It's not a matter only for ISA format and metabolites, but also all the different repositories. We need to agree as a community to a set of terms that can really make our life easier once we want to move to the semantic web representation. So one thing I would like to mention in the last 2 minutes I have is that we've been how to engage with ISA community and help the metabolomic community to build better tools. So I will show you the experience that we had the last few days. We were in Hong Kong with people at the BGI, Scott Edmonds, Rob Anderson, and also Ralph Weber from Birmingham, Reza from Metabolites. This was part of a BBSSA UK-China Award and BGI-funded hackathon. And there we developed the code that I've just presented, the ISATAB web viewer. We also developed functional specification and started coding towards a design of experiment wizard that would support programmatic creation of these ISA files based on just relying on the information of the experimental design, just to help you template these data structures very early in your process. Don't wait until you're about to publish to track the metadata. You should intervene much earlier. It's like a disease. The earlier you intervene, the better the outcome, really. So it's disease management. And we had a lot of fun. That was really fun. This is, I have to really be thankful for the people at the BGI, Peter Lee, Scott Edmonds, where is he? Here, Jesse, and Ralph. Yes, all very helpful people. And basically, what is good? The good news is that there will be a second hackathon. Again, we've got funding left. So if you would be interested, please get in touch with us. We can contact us here, and we will be very happy to see how we can get involved and get more feedback from you. So one last thing, in the 30 seconds that I've left, don't miss out. Today, there are two people, very important people from two very brave publishers which are moving into the data journal field. So there is Scott Edmonds from Giga Science and Susanna Sansone from NPG Scientific Data. They're also obviously the representative of the metabolomic repositories. So if you need help to deposit data, or if you want to discuss and engage with us, just grab us and we will do our utmost to help you to do that task. And I will stop and take any questions.
Excellent presentation. Of course, we're all for open access, no-brainer. From the metabolomics data sets that are out there, available to the public, I guess how many of them have proper metadata, and how many of them were successfully downloaded, used for further analysis, and resulted in new insights or publications?
Okay, so I guess this question is also mostly for Reza. So my understanding is that there are about 78 public data sets at the moment. We've managed to download them, so the conversion to RDF, the task that we were using, we managed to convert 80% of them. We can read them in R using the RISA package for most of them. Then the question about the reusability, I don't know. What I do know, however, is that we can do better in terms of annotation. People tend to be a bit lazy, I would say, and I guess we can. So maybe because it's too difficult, but we can enhance the other kind of annotation and provide more.
You talk perhaps all the time, but from the metabolomics community and the bioinformaticists out there who are interested in accessing metabolomics data sets, right? Can they easily download? Do they have all of the metadata? And can they really do what they need to do? Is my question.
I think one is interesting would be also to hear from you. I think that's the kind of main message. What are the queries you would like to run? And if you run the query, where does it fall?
There are
Have you
downloads.
been successful as a community?
Very, very small, to be honest. Compared to the proteomics that been around for a while, I mean, there's a proteome exchange, there's a data resources, and they recently announced, and there was a nice presentation by them, that actually people reuse the data set and generate new knowledge from existing one. We are still a long way towards that. I know that a lot of studies are being downloaded and used. They can see they're downloaded and used in log files, and I have personal communication with some of them that leaves the data sets and communicated back asking for more data. So there are data sets that are being used. I think one of the data sets, happened to be mine, have 5 times been used, and 3 of them are in new publication, but this has been over a very long time. The newer one is still a lot of these data sets has been submitted, gone online for the past couple of months. Still would take time to actually generate, but this is the aim. We have to have the right foundation, and we are learning as well. It's not that we are a perfect system of capturing. We make mistakes, we learn these things. That example that Felipe shows is actually one of our mistakes. Maybe partly being responsible, but we are learning, and we want feedback. We provide workshops. We have a lot of announcements via Metabo News. We go to the PSI community with Stefan, and Stefan is not a partner. We were 2 of us, but there are 80 people that are programmers, writing software, validated tools, packages. We are still a long way to get that, and we are trying to encourage people to get these workshops that Phil announced and other ones, and Tab and others. We are trying to get people more involved, and we track and report back how this information is used. But I guess this opens a discussion, so anything, if you want, we can continue with the discussions. Or Stefan, do you want to come here as well and contribute? There's a microphone, free microphone here for you to.
So just as a direct answer to that, I know that my study, which you find the URL on the back of the T-shirt.
We give T-shirts as well if you submit.
So that study has been used in teaching because it was used in teaching in a lab that is doing bioinformatics and doesn't have their own metabolomics facility. So it's an open data set out there, so I used it for that. The other thing that we've done with the metabolites team was to actually run all the mass spec experiments they had through XCMS to generate some overview pictures of that, which of course you only can do if you have open data formats, just to repeat that one again. That is also a way to validate that we have a pool of stuff. So we need a critical mass of data in metabolites to see what's the best practices out there. So the reason that the manual is very brief at the moment and not really giving you many guidelines is because metabolites people want to see first, what are people actually doing with the repository? What are they using that for? And it's now, after the 2 years that it's in existence, that patterns emerge, and we see that what the community is doing with the repository, so we can kind of not prescribe, but suggest the best practices for a really gold study in metabolites.
We need these success stories to encourage people to deposit and see the value. So if there are 10, 20, 30 bioinformaticists who are working in parallel, or can they download, can they do something, how can we motivate, how can we remove the bottleneck, and instead of saying this is going to take a long time, make it shorter? Maybe, I'm just wondering.
I mean, there are success stories, but proteomics has success stories, and genomics, but hopefully we'll get there.
I was just going to answer the point, Reza, in that, just like you say, there are some nice success stories from the microarray community where you can sort of say, well, somebody's asked you to do some RT-PCR and you don't have the money then to go and do the RT-PCR. You can go and look up that microarray study that's been done on the species that you're interested in and say, well, actually, somebody's already done this, and I've got the expression profile. It's there published in Geo or Array Express, and submit that data. I think the problem at the moment is we don't have enough data to do that with the submitted data sets at the moment in terms of cross-comparing across species, but it will come, I think, in the future. Another exciting area is that as certain formats for acquiring data become more popular, so we've been doing some work on Biocrates data set, and we've got about 7,000 samples that have been run through the Biocrates kit. We can begin to then trade on human studies and say, well, we'll have 11,000 people here. We reckon we can get significance from that, but if we can combine with a study that's got another sort of 5,000 or 10,000, you can begin to do these large-scale epidemiology studies that we see being done by GWAS, possibly through the repositories. I think there's a lot more work to do in terms of how you get that human data open access and what you can make open access for the human data. But I think we should begin to see these data sets traded, particularly for model organisms like yeast, where people have been trading data for some time.
Well, I could also add that in the last 2 days in the hackathon meeting, there were three tracks, in fact. 2 tracks were coders, and another track was people hands-on coding with ISATAB datasets. So really, we managed to collect about, I think, 500 samples worth of data in a few hours. So that was extremely efficient. I think this venue should be the case. Again, join us, tell us if you have data sets that you would like to submit. We can definitely help you and show you around. It's not that difficult. If you have your data in spreadsheets already, it's a matter of a couple of hours max. We can really help you.
Just to add on to what Jules was saying, until you get several hundreds of data sets in there, you're not going to have enough where you have comparable data sets. And the question is right now is, with the limited number that are there, how heterogeneous is the metadata?
Well, this is a very good question. I think, and this is why also we run this challenge of conversion to RDF, just to see, because it was a very good way to test. It was an acid test to see if we could run queries that were meaningful. And of course, it indicates that we need to do a better job. Stricter curation rules need to be enforced. We need to agree on vocabulary a bit better. In some areas, we have list of terms, but they are still not necessarily vetted by the community or present in the tools. So we've been working, for instance, with Biocrates as well, getting examples. They've got their own vocabulary, but we need to do this mapping exercise. And it's always a bit of a waste of time, this mapping exercise. So if we can decrease that, that would be extremely beneficial. And we could definitely document this curation process now, because the number of datasets is fairly small, but it's a very good lesson already to identify which bits are critical and which bits are maybe less so. And also what is extremely important is that we need to get the query case correct. So we need this interaction with this dialogue with the community to tell us how we like to query the databases. Of course, we can log your queries and we can try to second guess you, but it'd be much better to try to engage that earlier so we know exactly what to capture.
How, I assume that species is captured reasonably well, but what about experimental conditions?
Oh, that's a good point. The experimental conditions, there is a strong emphasis to describe. You mean the treatments? You mean how the things have been perturbed? And yes,
yeah.
Indeed. This is the kind of thing we try to focus, and we really try to harmonize things. So if you report a toxic exposure or a stressor, we would like to always have reported the nature of the stressor, the intensity, the duration, these kind of things, or the repetition, number of cycles that you have applied. And again, this has to be documented. People need to get used to this way of structuring information. But that's the only way to really make the most of the annotation. And it pays off in the long run, definitely.
So that was a very interesting presentation, and I just wanted to raise the third part of your sort of pipeline, as it were, that you didn't mention a lot of detail, which is the use of the semantic web, and which you, if I read you correctly, you see as some sort of goal that should be tried to be attained. It strikes me what you've done with ISA is almost where you need to stop in a way, because I mean these discussions about the use of the semantic web and marking up everything, and I think the exchange that just took place, if you look at GEO, yeah, there are thousands of studies submitted in there, and some of them have very good markup, a lot of detail about the experimental conditions, and some have none.
Yeah.
And do you think that extra effort is actually worth it, or it strikes me what you've done is incredibly sensible, because it's an ideal trade-off between having reproducible data, the metadata that can be used for both documentation purposes and analysis. Mm-hmm. But it's something that everyone can use, as opposed to pushing everything into this kind of XML, all these markup languages that are just difficult for most of us to use.
In a way, that's a difficult point. In a way, the table is to lower the barrier for people to open a document and start annotating. However, we cannot rule or deny the fact that, especially with platforms such as Biocredits or Metabolomics, has potential to process thousands of samples very quickly, I guess. And then obviously maybe the tables are not the best tool of choice for this super large experiment. So we need to find better structures. However, this is at the moment owing to the size of the experiment where we can go up to 1,000, 4,000 samples. That's okay, that works. But keep an eye on the long term and those big studies. And at the same time, what I think what is difficult for, or what maybe let down some of the scientists on the curations, on the query side of some of the big repositories, at the moment you still need to download a bunch of studies locally, filter them out, process them locally, discard, and then everybody has to do the same task. I think that's still too expensive. We need the better ways to cut, slice, and dice those huge data sets on the repositories. And I think some of the hints that we have from the storing that in graph database provides a better way. But that's just very early days for me. Yeah.
It's a very quick question because we're just running out of time.
Okay. Please.
Well, I was just going to add that, I mean, Oliver Fien introduced the concept of this ring trial in the earlier session, and it seems to me like that's a perfect marriage, not that one in particular necessarily, but just international efforts of ring trials doing or reproducibility trials that are then connected to the databases and analysis. It seems like that would be sort of a calibration point for a lot of what you're talking about.
No, absolutely. Unfortunately, I was not at the session, but definitely, yeah, I think that sounds very exciting.
Yep. Yep. This will be good to make this data available so people can actually test. Okay, with this, I want to thank all the speakers and thank you for your listening. We are here. Just find us and please get in touch. Thank you for coming.